Hello, everybody. Thank you for joining us today in our webinar on the use of AI in neuroimaging. Huge thank you for our wonderful panelists for taking the time to share their expertise with us. I also want to thank the rest of the OHBM-AUS webinar committee that helped put this together, especially Dr. Matthew Greaves and Dr. Natasha Taylor. Before we begin with the talks, I just wanted to give you a quick run through of today's webinar. So each of our three speakers will present the work for about 15 minutes, and then we're going to have a joint Q&A session at the end. So I'm going to ask everybody to write down the questions and hold on to them until after all of the sessions, so we can ask them all together at the end. Okay, now to start us off, it is my pleasure to introduce our first speaker, Dr. Forough Habibollahi. Forough is currently working as an applied researcher and postdoctoral research fellow at Cortical Labs, where she's working on creating biological intelligence by fusing living brain cells onto computing devices. I believe she's one of the people responsible for the neurons playing Pong that you might have seen, but I'll let her tell us all about it.
Hi, everyone. Thank you so much for the introduction and for the invitation. Yeah, I guess I could call myself one of the people responsible for making that happen. Hopefully, we'll try to make even more happen soon in the near future. And I'll just start sharing my screen now for today's talk. Basically, I'll be giving a overview of basically what we do at Cortical Labs, what our technology is, how we're building it, and what are some of the applications that we have thought about so far, and also, as much as time allows, some of the more research-oriented projects that we're doing at the same time. So I'm assuming you have my screen all right, and you can see my slides. Yeah, thank you so much. Yeah, thank you. So as already mentioned, I work at Cortical Labs. We're a startup in Melbourne. For those who may not be familiar with our work yet, our work basically sits at the intersection of neuroscience, synthetic biology, and to some extent AI. And basically at Cortical Labs, we're not just inspired by the brain, but what we're trying to do is to actually interface with it directly. So we all know that it's been a couple of years that we've been living through this extraordinary time in artificial intelligence with large language models that are really pushing the boundaries of what machines can do. They can understand language, they can generate very coherent videos now, which is all very impressive, but it's also raising deeper questions for us now. Like the main one could be, what is intelligence really? And can we build systems that learn and generalize really as flexibly, efficiently, and robustly as the human brain? While today's AI is undeniably powerful, but we all know that it's also data hungry, it's energy intensive, and it's also often very brittle when it comes to generalizing across different tasks or data sets as well. And then this is where this interdisciplinary field comes in, basically a growing field that sits at the intersection of artificial and biological intelligence. And at Cortical Labs, what we're doing is that we're building on this idea, not by just simulating the brain, but by interfacing with real neurons and directly leveraging their inherent powers of learning quickly, efficiently, and adaptively. So basically, we explore what it means to compute, learn, and adapt using living neuronal networks. And then eventually, we want to know what insights this might unlock for us about the future of intelligence or the meaning of intelligence. So as a quick introduction to the entire system and what we're doing there is that we're trying to introduce a new kind of computing paradigm. What we do is that we use human-induced pluripotent stem cells, or HiPSC cells, to grow real neuronal cultures on microelectrode arrays. If you're not familiar with these cell types, basically this is like a technology where you can get a simple sample of skin or blood donation of volunteers, revert it back to stem cells, and then grow it into whatever type of human body cells you want, in our case, to neuronal cells. Then these are cultured onto microelectrode arrays. And here, for example, you can see that these are actual microscopy images of some of our cultures. On top, you can see a 3D neuronal culture or organoid, and on bottom, it's a 2D or monolayer culture. Then these networks start to self-organize, connect, and actually fire in very rich, spontaneous patterns when they reach a certain maturation point. Which basically means they're not just alive, they're also reactive, plastic, and under the right conditions, we've seen that they start to show learning-like behavior. Yeah, sorry, this is the microelectrode arrays, and then what we've seen is that if we treat these neurons as programmable systems, not just by recording from them passively, but stimulate them, record from them, and then in a closed loop feedback system, try to teach them tasks, actually they start to show learning-like behavior. So that's exactly what we're working on, a closed loop system where the neurons adapt based on the input and the feedback that we provide to them over time. And we've seen that things like changing in firing pattern, connectivity, and even task performance metrics starts to appear in the system. So we call this CL1, which is basically short for Cortical Labs' first generation living processor. You can think of it as like a wetware processor. So it senses input via stimulation, processes that input through biological computation, and then expresses output via its spiking patterns. So it's alive, but it's also engineered for experimental control and computational interfacing. What we have now is that apart from that, the device itself or the wetware processor itself, there's also option for cloud access to it. So basically, while we're maintaining these units, you could be accessing them online via our cloud-based system. So this is just an overview of what one might be seeing there on the system. So these are actual channels, then the spikes that are being detected on these channels while either the culture is just showing their spontaneous activity or they're going through a certain task in our closed loop environments. So yeah, we're trying to imagine a day where we can have these racks of all of these units potentially interconnected as well, and try to start using them as these new biological and live computing systems. So with these CL1s, again, we're not just trying to record the spikes, we're studying how the network of these neurons processes signal over time. We look at how different stimulation protocols change these networks' connectivity, information routing, and population coding, just like you would do in a synthetic or artificial neural net, but here it's just real biology. For those who may be familiar with our first work of the game of Pong, this is kind of like an old visualization, but I feel like it helps a lot in understanding what we mean by closed loop feedback system. Imagine we have this virtual environment of game of Pong that you can see on the screen. And this is kind of synced and linked online with our neuronal cultures. What you see here is actually a multi-electrode array with all the electrodes and the activities that you see on top of electrodes popping up. I'll just repeat this. What we do is that we predefine these regions as sensory, for example, on bottom, and then two motor regions on top, each of them responsible for one direction of the paddle movement in the virtual environment. Then we feed the information about the location of the ball in this case to the cultures via place coding and rate coding. For example, the x-axis location of the ball, or how far it is from the paddle, is encoded as the frequency of the input. So the closer the ball, the higher the frequency. And the y-axis location, we just divide the sensory region and the y-axis of this virtual environment to equal segments. And whichever segment on the y-axis the ball falls within in that particular point in time, we stimulate only a certain segment of our sensory region. So basically place coding the y-axis location. And then using the activity patterns from the two model regions in kind of like a competition, whichever model region wins, the paddle in the virtual environment moves to that direction. So predefined directions are assigned to these two motor regions, basically. And then if the ball is hit accurately, there's a very short period of reward. And if it's missed, there is a longer period of punishment. And for punishment in our initial work, the Pong paper, we used a random unpredictable signal with random frequencies at random locations in the sensory region, which showed to be actually helping the cultures to show learning-like behavior and improve their game performance metrics. So that's just one of the examples. Again, the earliest example of a task environment or a closed loop system that we tried. Just to show you what we're doing now, we're moving beyond that now. We're trying to design other game environments or task environments. This is one that Moin is designing now or has finished designing. It's a Flappy Bird game. So basically, we're trying to control the height of the bird using the activity from neurons and try to basically keep it as close as possible to that target line and punish it when it hits the ceiling or the floor. And obviously, we expect to see more and more time spent near that target line as the cultures start to learn. But then again, I want to show 3 more examples of other types of tasks that we can do and we're doing these days with these cultures. We call them classification tasks. Basically, each of these tasks, there are 3 of them, they're increasing in complexity and difficulty as we go on. So in the first task, our goal is to teach the culture to distinguish between 2 distinct input signals that only differ in their spatial origin. We call this a binary task. So imagine as presenting symbol 0 or symbol 1, but we just encode them in actual location of the stimulation. So then we randomly stimulate one of these 2 pre-selected electrodes across many repeated trials, and then using a very simple linear classifier, we assess how accurately we can decode the culture's response and assign it to the correct input class. And for example, this plot shows that the classification accuracy for the actual neuron responses is much higher compared to random spike controls. In the second one, what we do is that basically repeat the same thing, but this time same location of the stimulation, only with different frequency patterns. We call it Morse code. And again, we expect to be able to very simply and linearly distinguish between the pattern of responses when either of these 2 frequency patterns are represented. And again, that's what we observed in our pilot data. And then finally, we increase the complexity 1 more step. Instead of only spatial or only temporal patterns, we use spatiotemporally encoded basically representations of digits. We use the very popular MNIST dataset, which is thousands of handwritten digits between 0 and 9. We use an autoencoder to encode every one of these digits into a spatiotemporal spike train, basically, because that's how we can communicate with our cultures. And then again, we try to classify the neuronal responses into 9 different categories or classes based on the evoked patterns. And what's really exciting is that we already see signatures of learning in these networks. And when they're exposed to consistent feedback loops in terms of whether they classified accurately or not, the neurons start to adapt their firing patterns, and they basically try to avoid or amplify specific actions. So that's why we start thinking that it's probably not just Hebbian learning either. What we're seeing is that we're seeing forms of plasticity that are dynamic and context sensitive, that suggest richer learning rules at play. So basically, yeah, we can shape the network's behavior and encourage it to associate certain stimuli with certain outcomes. Now, in the second part of my talk, I'm not sure how much time I have left, but I want to very quickly go through two of the more recent research projects we're doing using this data set. One of the most recent ones is what we call a Tavern. It's a method. It's a graph-based representation learning method of neuronal dynamics and behavior. The method was primarily designed by Moin. And basically, this is a model to try to find lower dimensional representations for a sequence of graphs over time. What it gets is at each time step, the model uses a graph neuronal network to capture the network's spatial structure, and then a recurrent neural net or an RNN with an addition of a temporal attention layer to learn how the structure evolves over time, and then it tries to capture the changing nature of neuronal connectivity. Why we designed this method was that basically up till this moment, in all of our previous publications, we were trying to see how the network pattern changes, how plasticity changes, what are the network features or characteristics that change during gameplay versus rest state or when the game is playing well. But we hadn't been able to actually associate the changing patterns of the neural networks to behavior before. So using Tavern, this is what we're trying to do. And one of our key findings, very briefly, this is obviously like a full paper, but please ask me questions later on. But using this method, basically what we did is that we found a lower dimensional representation of the activities of every single channel or electrode that we have in different windows in time during our gameplay and rest sessions. Rest is just a spontaneous activity of the cultures. And then for each of these time windows, we also know how well the game was being played. So we have the game performance metrics, such as hit to miss ratio. And then we rank these windows from high to low. You can see on this figure from left to right. Basically, left to right is decreasing game performance in that particular time window. So for every time window, we first build the functional connectivity network among the spiking time series of the channels, feed that into the Tavern network, the Tavern framework, and then get these representations in a lower dimensional space that we can visualize like this. So every dot here is one of our electrodes or one of our channels, and the colors actually show which predefined region these are associated to. So for example, the light blue is the sensory region, then we have purple and pink for the two basically halves of motor region, which we have pre-assigned to be responsible for movements up. And then yellow and green, the two motor regions which we have pre-assigned for paddle movement down. And what we saw was actually very interesting. We saw that during rest, the network remained relatively diffuse with no clear structure or change across time. But during gameplay, especially in the top performing time windows on the left, we saw that distinct clusters start to be formed. So specifically, sensory channels make their own clusters, and motor channels associated with the specific movement direction begin to group together in a meaningful way as well. So what's even more fascinating here is that some of these motor channels, although they're functionally similar and we've assigned the same role to them, they're actually physically not neighbors to each other on our electrode array. So they become very tightly associated in their activities when the culture is actively engaged in the task and is being successful in the task, while they're actually very far from each other physically. And remember that we pre-assigned these roles experimentally without any prior ground truths. And the fact that these specific clusterings start to emerge only during successful gameplay suggests that the system is actually organizing in a task-specific and non-linear way. And this can again reveal some evidence about the plasticity and coordination that goes beyond just a spatial proximity. I'll just skip through this quickly. This just shows the evolution of a few samples of these cultures in time as we move from less successful gameplay windows to more successful windows. You can see the clusters start to emerge. So this was actually a very nice finding for us, and how we could associate some patterns and reorganizations of the networks to successful versus unsuccessful behavior. In another project that we're doing and looking at these from a very different angle is that we basically trying to dig deeper into how information flows through these networks now during these successful gameplay sessions or during rest. And for that, I'm gonna again very briefly run through this, but what we do is that first of all, instead of building just a functional connectivity network, we build a causality, basically causal Granger causality network, we can call it. So here the edges are not just correlation based, they come from a Granger causality analysis done between every pair of channels. Basically, this allows us to identify statistically significant directional relationships between the activity of different electrode pairs. Essentially asking, does knowing the activity in one of these electrodes help us in predicting the future of the other electrode? And if yes, there is a directional edge between them. And the more significant this is, the stronger the edge becomes. So here you can see just an example of rest versus gameplay and these causal connections. First of all, obviously, you can see how much denser this network is during gameplay. And then after building these networks, I also do one more step. You'll know why later on in the next slide. I also identify the most influential and influenced nodes here. Basically, influential being the nodes which have the highest number of outgoing links or edges, and influenced being the nodes with the highest number of incoming edges. And then after I am making these networks, identifying these nodes, I'm not gonna go into the details of the next steps, but basically I do some state space reconstruction. I find an optimal embedding dimension and an optimal time lag, looking at the evolution of these dynamic networks in time, and trying to make a trajectory in time for them. As we know, it's called a state space. And then using these state spaces, we can build these recurrence maps from them. I think I have, sorry, I'll do this quickly. I have hidden that slide, but I kind of feel like it helps a lot in understanding what I'm gonna say next. So I'm so sorry for this. Okay, so these are the recurrence maps using the reconstructed state spaces. Again, an example of during gameplay, an influential node versus influence node. These basically have time indices in both X and Y axis. And every dark point shows when the system has revisited a similar state. Sorry, I just realized I've passed my time limit. I'm just going to go quickly through the last slide. Basically, what we found was that during gameplay, we have high recurrence, high determinism, and high laminarity, or visiting the previous states, versus rest. But also something that was very interesting was that the influential nodes were actually the ones that were acting more closer to a chaotic state compared to the influenced nodes. So yeah, I'm so sorry. I completely missed the time and the notifications that are received from the hosts. So yeah, basically our neurons can perform inference. They show learning-like behavior, and it seems like the nodes that are near chaotic and show cyclic dynamics are the ones that are driving behavior in these cultures. Thank you so much to all of our team and collaborators, and sorry again for going over time.
No problems. Thank you so much for this excellent talk, Forough. Yeah, just a reminder to please hold on to your questions until the end of the sessions. Our second speaker today is Associate Professor Heath Pardoe. He is the Neuroimaging Theme Leader at the Forough, the Stream Lead for Science and Operational Support in the Australian Epilepsy Project, and works as part of the Epilepsy Imaging Group. He's also the co-director of the Forough National Imaging Facility Node and holds honorary positions with New York University Grossman School of Medicine, Monash University, and the University of Melbourne. His current work focuses on applying different computational analytical tools to improve our understanding and treatment of epilepsy. And he's going to tell us all about it now. Thank you.
Thanks, Priscilla. Are my slides displaying correctly?
Yeah.
Yeah. Okay, cool. All right, so thank you for the invitation to present. And that was a really fascinating talk that we just watched. It's quite different to what I'm going to be talking about. So today I'll talk to you a little bit about socioeconomic and disease-related correlates of brain health in epilepsy. And to measure brain health, we use brain age prediction. So for this talk, I'll give you a quick overview of the Australian Epilepsy Project. It's a large study that we're running here in Australia. And then I'll talk about how we use that data to look at the relationship between neuroanatomical changes and cognition. And then I'll give you a little bit of an overview of some work that we've done using the Enigma epilepsy data set to see how socioeconomic factors can also affect the structure of our brain. So the Australian Epilepsy Project, the chief investigator is Professor Graham Jackson, who's an epilepsy neurologist here in Melbourne. It's been running for the past, the main project's been running for the past 4 years or so. We're established at 7 scanning sites across Australia, which you can see there on the map, covering all the states. And we've scanned more than 1,400 participants. What we're interested in is epilepsy. And there are three cohorts of individuals that have had seizures, which is the main feature of epilepsy. There are people that have had a first seizure, but are yet to be diagnosed with epilepsy. And about 50% of those people will go on to get a diagnosis of epilepsy. About 1/3 of the cohort are people with newly diagnosed epilepsy. So is it within 6 months of their diagnosis, and about half of the cohort are people with drug-resistant epilepsy. And the key feature of this is that most epilepsy imaging research, of which there is a lot, is focused on that 50% of our cohort of people that have drug-resistant epilepsy. We really know very little about what people's brains are like before or when they've just had their first seizure. And in fact, one of the main questions that the Australian Epilepsy Project is hoping to answer is that we know from population-based studies that 2/3 of people with epilepsy get on the right medication and get their seizures under control and can get on with their life. But there's this 1/3 of people with epilepsy where medications don't work. They have what's called medically refractory epilepsy, and their life really, they really suffer for that. Their quality of life drops. And at the point of diagnosis, when a person's sitting across the desk from their neurologist when they've been diagnosed with epilepsy, there are no markers that can tell the neurologist or tell the patient which group they're going to fall into, whether or not they're going to continue to have seizures or if they're going to find the right medications and stop having seizures. And so that's a question that both ourselves and many epilepsy researchers across the world are trying to address. So we collect MRI scanning data, obviously, as part of this study. But for anyone that works, doing AI or machine learning based analysis of MRI data, really, the acquiring high quality MRI scans is only half of the effort. And really the additional data that you collect that's not imaging data is equally as important for training a useful predictive model. And so as part of the Australian Epilepsy Project, we collect a detailed clinical history that describe the person's epilepsy. We collect cognitive data, which I'll talk about in more detail later. We have an advanced image acquisition, takes about 90 minutes in the scan, and we also collect genetic data. And we track these people for 2 years after their baseline assessment. And the reason that we do this for 2 years is that's the length of time that it takes to determine if someone's going to be medically refractory or not with a high degree of confidence. Progress to date, the primary AEP site's based in Melbourne, so you can see the majority of participants have been acquired here, but you can see we've been scanning people from all over Australia. Okay, so just to give you a quick overview of the image acquisition protocol, we've got our standard T1, whole brain T1 weighted and FLAIR acquisitions, which is the imaging data that I used for the analyses that I'm going to describe later. We also have for people with epilepsy, most of the time the seizures come from the temporal lobe. That's a general sort of principle. So we have some image acquisitions that are very targeted to the temporal lobe. We have a T2 weighted scan that gives nice high resolution images of the hippocampus, which is a critical structure in epilepsy. And we also have an acquisition that's designed to detect a pathology that's called an encephalocele. It's not the topic of this talk, but it's very interesting. This is when there's a defect in the skull and the brain tissue sort of gloops out through the defect and then, is not where it's meant to be and gets irritated and is a cause of seizures. And so that's what that sequence on the bottom right is designed to detect. And this is a relatively new pathology that's been identified because it's been very difficult to detect on standard clinical MRI scans to date. And so interesting findings regarding the incidence of temporal lobe encephaloceles in people with epilepsy. We also obtain QSM or SWE scans. We do whole brain T1 and T2 mapping. The participants do language and working memory fMRI tasks in the scanner, and there's also a resting fMRI acquisition where the participants watch segments from 3 films. The first is the social network, which is very dialogue heavy, so drives those associated networks in the brain. Another segment is shown from the movie Inception, which has a lot of visual effects, so activates visual networks. And the third movie is Ocean's 11, which features a lot of celebrities. And as it turns out, that really activates specific networks in the brain that are oriented towards facial recognition. And it's quite agnostic to a person's cultural background. Turns out celebrities are famous all over the world. So we also collect multi-shell diffusion weighted imaging data. So that's the image acquisition for the Australian Epilepsy Project. But as I mentioned previously, my work is focused primarily on whole brain T1 weighted and FLAIR acquisitions. So just to give you a quick background on how we can use MRI scans to predict a person's age and how that might be associated with an individual's brain health. Our brains change as we age structurally and functionally. MRI can be used to visualize and measure these age-related brain changes. And there's been, over the last 15 years or so, there's been a whole suite of AI or machine learning-based methods that can use as structural MRI scan and functional to estimate the age of a participant. And if we take this MRI-based age prediction and subtract the person's chronological age, we get a measure that we call the brain predicted age difference. Terminology can vary across studies, but at the end of the day, this metric is our candidate measure of brain health. And there's good reason for us to make that link, which I'll get to in a minute. So we're probably all quite familiar with FreeSurfer by now. It's been around for longer than 25 years. In brief, for modeling the surface of the cortex, this is a software package that delineates the boundary between the white matter and the overlying cortical gray matter, shown in yellow, and the outer cortical gray matter shown in red. And if you measure the distance between those two surfaces, you get the thickness of the cortex. If we have a look at how the thickness of the cortex changes over the human lifespan, you can see it steeply decreases up to the age of about 20 or 30, plateaus for a little while, and then after 50 slowly declines. So you can already see from these graphs that if you measure the thickness of the cortex using an MRI scan, you can infer the age of the individual that was scanned. And these machine learning methods really work off the same principle, with the caveat that the shape of this curve varies depending on where you are in the brain. So you can get more accurate age estimates than shown from this graph just due to the spatial nature of cortical thickness changes and that variability. So these are previous studies. The graphs on the left show differences in white matter volume between people that are obese and people that are skinny. There's a shift to the right for people which are obese, shift to the left for people that are obese, and that can be used to infer this brain age gap. And it turns out that people that obesity is associated with an increased brain age gap from midlife. Quite a depressing plot shown on the right. People with an increased brain age gap die earlier. So there's an increased mortality risk associated with an increased brain age gap. And so these studies really illustrate that link that I was talking about before between an MRI-based brain age gap and brain health. And the higher the brain age gap, the higher is associated with poorer health outcomes. So we looked at this and have been looking at this in epilepsy. So for this work, we used the great thing about these brain age prediction methods, as I mentioned before, there's a variety of models to choose from. For this work, we used a model that was trained by Andre Arimia at USC. He trained his model on around about 5,000 participants. We applied his age prediction model to 182 healthy controls and nearly 300 epilepsy cases, and looked at the relationship between the brain age gap and measures of processing speed and memory that were done as part of the Australian Epilepsy Project. In addition, we looked at whether or not a person has an epileptogenic lesion or not affects the brain age gap, and also the number of anti-seizure medications that the person takes. And this age prediction model is a deep learning model that's based on convolutional neural networks. So just as a slight diversion, this is a tool that's developed as part of the AEP to do cognitive testing. You can see the AEP cognitive lead on the top left, Chris Tailby, running through the program with his postdoc, Jody Chapman. And these are some trail making tests, I believe, that show how cognitive testing can be administered remotely as part of the Australian Epilepsy Project, which is really critical for being able to reach people in remote and regional Australia. It means that to participate in the study, the only in-person or bit that requires travel is going to the MRI scan. All the other study activities can be done remotely. You see, here's a list of the cognitive testing data that's acquired, and the measures that we ultimately use, processing speed and memory, are derived from this suite of tests. So back to the imaging data, when you apply an age prediction model to your own data, a crucial step is to make sure that it's a valid model for your data. And so you really need your own healthy controls to plug into the age prediction model. And that's what that scatter plot on the left shows. For healthy controls, you'd expect on average, their MRI predicted age to be the same as their chronological age. And that's what this graph shows, that they lie along that one-to-one line. The violin plots on the right show the brain predicted age difference in each of our subject cohorts. And the only statistically significant increase was observed in people with drug-resistant epilepsy, which is shown on the right. And so these are people with chronic medically refractory epilepsy, and they have an increased brain age gap of about 4 years. So that shows that their brains resemble people that are healthy controls that are 4 years older than they are. If we have a look at the relationship between cognition and the MRI-based brain age gap, you can see that there's a negative relationship observed for both processing speed and memory. So people with an increased brain age gap have reduced processing speed and reduced memory. So there's a link between brain structural changes and cognitive changes in these participants. If someone has a lesion observed on their MRI scan, which is the case for about 20 or 30% of our cohort, that is weakly associated with an increased brain age gap. And if someone is taking a number of anti-seizure medications, both of these things are markers of drug-resistant epilepsy. That's also associated with an increased brain age gap. So we're really starting to be able to unpick the various factors that can contribute to an increased brain age gap in people with epilepsy. To shift gears slightly into, I'm just on the last couple of slides. This is not AEP work. This is work that we did with the Enigma Epilepsy Project, using a different brain age prediction model, a slightly older one, but trained on a larger number of subjects, 30,000 cases. And being Enigma Epilepsy, we're able to pull data from a number of international sites. So you can see that we're really starting to get decent size sample sizes, so 1,200 healthy controls and 2,600 epilepsy cases. The finding that was really compelling from this work, because we used cases that were taken from all over the world, we could correlate the national GDP per capita in our participants. So that's a measure of socioeconomic health, if you like, with our MRI-based brain age measure. And you can see, surprisingly, or perhaps unsurprisingly, that there's a negative relationship there. So if people come from poorer countries, there's an increased brain age gap. And that's equivalent to a 1-year decrease in brain age gap per $20,000 increase in GDP per capita. I think something to note is that even though we have a good spread in GDP per capita countries that contributed to the study, they all fall on the wealthier side of the spectrum. That's what's shown on that bottom left graph, but quite an interesting finding nevertheless. So just to summarize, drug-resistant epilepsy is associated with a deviation from typical aging trajectories in the brain. A higher brain predicted age difference corresponds to poorer cognitive performance. In drug-resistant epilepsy, a higher brain predicted age difference is associated with both anti-seizure medication use and if you have an epileptogenic lesion. But it's important to bear in mind that brain predicted age difference reflects more than just disease. That the final couple of slides I showed you suggest strongly that socioeconomic factors also play a role in age prediction using MRI. So thank you for your attention. The AEP team's very large, and this is the team here. Thanks very much.
Thank you so much for your talk. Just in the interest of time, people can start submitting their questions to the Q&A portion of the webinar, and then the speakers can start answering as well. To finish us off, our last speaker is Associate Professor Zhaolin Chen. He is an Associate Professor at the Department of Data Science and AI at Monash University. He's also an ARC MCR Industry Fellow, and he provides leadership in imaging analysis at Monash Biomedical Imaging. His current work focuses on improving neuroimaging acquisition, analysis, and reconstruction using multiple computational tools.
Thank you for the nice introduction, and thanks everyone. Am I showing the right slide? Right view? Is that correct?
Yeah, perfect.
Okay, excellent. Thank you. That was great talks from previous speakers. I'm going slightly different angle. So my research areas in the image reconstruction area, image generation. So my talk will be around the inverse problem and the relationship with generative AI, which is a recent major development in the field. And also I'm going to show a couple of examples. One is around the ultra low field MRI. Image enhancement, we have been working on this for a couple of years, and some other image analysis applications. So I'm going to talk about a little bit background on the image reconstruction and the synthesis work. So I'm just showing the medical imaging workflow. So as soon as you have a participant and a subject coming into the scanner, well, very importantly, we measure the raw signal, MRI case space and PET-lismal data, and then we go through image reconstruction to calculate the image, to generate the image for visualization, interpretation, and also we can do subsequent image analysis to identify markers such as segmentation, classification. So from the signals and systems point of view, let's say if we have a ground truth subject, P of F, come in, and there is a forward model, which is G, as the imaging system is a function of hardware and software. The hardware will be MRI coils, the detector, PET detector geometries, and software will be, for example, MRI pulse sequence. And then we have a measurement space, M, and then the inverse model to calculate from the measurement space to back to image space would be the inverse model, H. Right, so when we do image reconstruction, what we want to do, we want to have an accurate representation from the reconstructed signal to original ground truth, we call. So the examples can be, T1, T2 structural images and various functional image as well. And in terms of accuracy, what we want is minimum noise, minimum artifacts, reduced bias, image homogeneity, and very importantly, quantitative accuracy, especially, for example, with PET imaging. So if we can do this everything perfectly, what we can do, we don't need deep learning, right? We can just invert the forward model. However, the problem is coming from uncertainty. Some of those are human-made uncertainty, some of the hardware physics-related uncertainty. For example, the imaging hardware, we know there's a gradient is not, when you call a gradient, it's not perfect. You have delays and the field homogeneity, and there is a fundamental physics principles we have to follow. And often, because we want to speed up data acquisition, we want to have undersampled measurements so we can reduce scans and motion artifacts, for example, was another uncertainty source. So all these factors will cause M to lose information from the ground truth and G become an ill-posed problem. Conventionally, we can still solve this. In past century, people have developed techniques to solving this type of problems. I'm going to skip a couple of slides here, considering time. Very importantly, I want to highlight the comfort sensing work to solve this type of problem, which are cornerstones for many of the deep learning works in this space as well. So the image is courtesy of Terence Paul, a well-known Australian American mathematician. So he looked into comfort sensing concepts for MRI, actually. So one example is showing here. In conventional least square, if you have sparse sampled signal, for example, this is a radial sampled K-space, this is a renal image. If you use the least square, you're bound to have image artifacts because of the aliasing artifacts. You're not sampling the signal according to accuracy rates. So there's causing problem. However, if you know the signal is sparse, as a second example, Terence is showing here, if you know the signal is sparse, you can optimize the sparsity of the signal by calculating the L0 norm of the signal, so which is the sparsity of the signal itself, and you can actually get a near perfect reconstruction if you don't have any other noise. However, this is an NPA hard problem. You can't really solve L0 norm. So a good approximation alternative is the L1 norm. So then you will calculate the sum of the magnitude of the signal to approximate the sparsity. Well-deserved credit to Mickey Lustig and John Pauly to implement, demonstrate in MRI in vivo for this implementation. So this is optimization problem essentially is used. So it's L1 norm of the signal. So sum of the magnitude of the signal and constrained by the data fidelity term. So the physics guided regularization of the signal. So the comfort sensing shows much better improvements compared to conventional techniques. And that's was seminal work in the field with comfort sensing. The reason I mentioned about comfort sensing is this scheme, optimization scheme, is used in deep learning. many deep learning techniques initially from the denoising type of approach to later on generative AI to signal recovery generation type of approach, still following this type of mechanism, optimization mechanism. So this is, I want to use this slide to show the evolution of MRI reconstruction techniques. You can see from the conventional, very early on, the voxel-wise reconstruction to the very important parallel imaging and regularization over well-known one is total variation. Regularization uses many fields. And then computer sensing, as I mentioned before, and you can see deep learning models starting from 2014, 15, 16, but of course, many works are done before that. So you can see the deep learning use a neural network to approximate the signal, whether it's for denoising purpose or image generation purpose, and subject to a physical constraint to make sure the data following physical properties. So I'm going to switch gear a little bit. This is the timeline of independent MRI, of course, for the generative models in medical imaging development. We did the review paper back in 2024. So you can see cluster of techniques start from UNet and the GAN family, cycle GAN, and attention guided GAN, and then moving to transformer-based techniques, starting from 2022. So that's attention aware and the swing transformer, which is a very popular backbone, and then moving towards to diffusion techniques, the denoising diffusion, of course, and the latent diffusion. And adversarial diffusion is proved to be very efficient and a very high quality technique. A lot of our work are using adversarial diffusion architecture. So generative AI can be used in many tasks, in translation tasks from MRI to CT, MRI to PET, low field MRI to high field MRI, different contrast translation and PET image reconstruction, and PET to CT image translation. So this was the plot that we showed that the different backbone models and cluster of techniques, the popularity being used in the field, and in terms of brain neuroimaging, considering the focus we have for today, you can see DDPM, which is the denoising diffusion model, shows pretty good mean square error compared to other early models. And in the synthetic MRI, for the brain application of the PSNR, and the DDPM shows pretty good structure similarity compared to the other generative models. So I hope this gave you a general trend of the backbone AI model and the relationship between the computer sensing and this generative AI-based model. I'm going to show you a couple of examples in the next few slides. So the first example is the ultra-low field MRI enhancement work we have been working on, and image and other image analysis applications, depending on the time, how much I can show. So the ultra-low field enhancement project was supported by National Imaging Facility. We've been working on this for past couple of years. We recently got a further support from National Imaging Facility to continue the program. And we also collaborating with others in different applications. One was a successful grant led by Karen from Deakin, and we're looking at the traumatic brain injury. The other grants with Alfred Hospital look at MS applications. So the initial project was looking at the stroke, and we have a quite wide range of data acquisition. One of the focus was to develop and apply AI techniques to enhance ultra-low field image, and also developing decision support for the clinical application focus. And our ambition really is to make MRI accessible, AI-empowered medical imaging for regional remote applications and patients living in regional areas. I'm going to show why we need AI in this application. So on one hand, the ultra-low field MRI has much smaller footprint and cost-effective, all the nice characteristics associated with ultra-low field, as you can see at the bottom of the slide compared to conventional slide. However, the main problem is the image quality. This is the example shows different contrasts from the 64 millitesla MRI compared to 3T MRI, which we often see very regularly in our studies. And the ultra-low field MRI shows much lower spatial resolution and poor signal noise. Sometimes the contrast is also lower, especially for T1 and the flare. And for diffusion MRI, you can see the contrast signal noise is much poor compared to the Synthesis 3T scan. So there's certainly a large room to further improve the image quality compared from the low field compared to the high field. I'm going to skip this slide. I'm showing you this is one of the model we developed. So this is the diffusion based model. By diffusion, I mean the deep learning based diffusion backbone model, not MRI diffusion, and using a adversarial guidance. So which proven to be very effective because diffusion modeling, you take many steps to degrade image in each step by adding small amount of noise. It's quite time consuming, but if you have a very good guidance from the critique model, and you can speed up this to substantially, and as well as having a better fidelity of reconstruction. So the results really showing the consistency across multiple sites. We have 2 sites which are healthy cohorts and 3 sites with disease, and you can see the PSNR structure similarity and mean square error are comparable and consistent across different sites. I'm going to show you a couple of clinical examples. In here, you can see the point of care MRI, although it shows the MS lesion in this example, but the definition of the location and the size, the sensitivity is much lower compared to the 3T. And in the model, deep learning model we developed the SimPOC, you can certainly see the improvements of the image quality as well as the identification of the lesion compared to the renal low field MRI. So this is the white matter disease, and this is the stroke. You can see the point of care, the boundary between the pathology and the neighbor tissues are less well defined compared to the high field, which is 1.5T in this example, and with the deep learning model, generated images much better recovered. I'm going to skip this, but maybe just quickly, these are the generated the whole 3D image. Okay, and of course, we measure the brain structure anatomical accuracy compared with the deep learning with and without deep learning models. You can see with deep learning models, the alignment between the measurements compared to high field is much improved by using deep learning model for various brain structures. So that again demonstrates the importance of using an AI model to improve the image quality for low field MRI. So next example is this is moving maybe very quickly. This is moving from a supervised paired the modeling to unsupervised, where we use 70 to translate the style from 70 to the ultra low field. So really mapping a 2 order of magnitude image quality difference. And we are able to show with the smart design of the model, in this we use implicit neural representation to improve the image. You can see the contrast noise ratio is much more improved compared to the original image quality. So this a lot of this work done by Dr. Tauhid, and this was published in Mikai last year. This is an example to show the comparison of the enhanced ultra low field in reference to 70 scan. Just very quickly, and we also developed a segmentation model that improves the clinical utility of these scanners. We can use this model to identify any anomaly from the low field generated image by using a smart segmentation tool. So this is work done by Dr. Himashi Pierce. So how much time do I have? Last 2 slides, and this is the large model we are developing. So we call it segment any tumor model. It's really trained by 17,000 and 11 public data sets to have a foundation model like segmentation model to for tumor application. So maybe this is interesting application of deep learning in connection with virtual reality. So we've been looking at how to, a lot of the segmentation models requires human in the loop approach, which is very important to make sure the segmentation is good quality. So this is the example of connecting segmentation with VR model, and we implement this in 3D slicer applications. All the models and everything is in there. If you have a virtual reality toolkit, you can also try in a virtual reality setup. It's quite interesting research area we're going into. I think that's pretty much all. Just want to acknowledge the sorry, I can't move to the next slide. Just acknowledge my my team and all the collaborators and the funding agencies. Thank you.
Thank you so much for the talk. I want to invite everyone to thank our speakers and for the wonderful work and amazing talks that they just gave us. Unfortunately, we're running out of time, so we won't be able to do a Q&A, but I would encourage everyone to reach out to the panel if you have any burning questions. Thank you so much for attending. We will circulate the recording, I think, to everyone that signed up as well after.